So the types of embryo sac.
Okay, madam.
The introduction part. During the maturation of ovule, one or more cells of nucellus, they develop into archisporial cells, or otherwise the megaspore makes the beginning of the female gametophyte generation. So the nucleus of the megaspore, it divides and develop into the female gametophyte. So in this, almost the female gametophyte of the angiosperms, that is very much reduced and totally dependent for its nutrition upon the tissue of the sporophyte. So the ovule, it is an integumented megasporangium. We know that. So they all are closed in the ovary. Almost we'll discuss in that the basal part of the ovule, where the nucellus and integuments and funiculus, they three were merged in terms of the collagen. So the ovules are oval in shape and the body consists of the diploid nutritive tissue. Okay, so it's nothing but nucellus. The ovules are the shape, it becomes the oval shape. So the body consists of the 2N or 2X status. So diploid nutritive tissue. So it's called the nucellus. The nucellus is surrounded by one or two protective layers. They are the integuments. We know that. So the depending on the almost number of integuments, ategmic, unitegmic, and bitegmic. We know that the three types. So in this, the region, it will present beneath the embryo sac, that is collagen, just to the angiosperm female gametophyte, that is critical for, so this is critical for plant reproduction. It is contains the egg cell and central cell, so that become fertilized and give rise to the embryo, so and endosperm of the seed respectively. In this, almost the female gametophyte development, it begins early in ovule development with the formation of a diploid megaspore mother cell. So this is very important. The female gametophyte development, it begins early in the ovule. So it will development with the formation of a diploid megaspore mother cell that undergoes the meiosis. So this is the complete development of the megasporangium. Just to, in this, almost the ovule, that is the primordium, appears as a small protuberance on surface of the placenta. So this is the first cell, Nana. This is the embryo sac mother cell, diploid cell. It will undergo the meiotic division. So it gives the 4 cells. So the 4 arranged in a linear shape. So in the 4 cells, the lower 3 were degenerate and upper one is the functional cell. So inner cell, it will grow as a complete embryo. Okay. So just we will discuss one by one in the development of the ovule. Okay. No, awesome. So the archisporial and megaspore formation, it's nothing but the archisporium is the hypodermal in origin at some early stage in the development of the ovule, usually at the time of the initiation of the integumentary primordia, the single hypodermal cell that is known as a primary archisporial cell. So it's simply, we'll discuss that the development, only 4 steps I will give the clear-cut information about the development of the megasporangium. So in angiosperms, almost sorry. So the development of the megasporangium in the almost in this 2 type of the tissues are there. So in the 2 crassinucellate and tenunucellate sporangium. So in crassinucellate, the ovules, the archisporial cells, they will undergo the periclinal division and forms an outer parietal cell and inner sporogenous tissue. So this is almost the female gametophyte, the collagen, nucellus, all parts, you know that. So nucellus, integument, antipodal cells, embryo sac, secondary nuclei, 2n state, so egg cell and synergids. So the egg cell and synergids both will form as egg apparatus, that is in the triple fusion 3n state. So this is the micropyle, this is the raphe, and this is the funiculus. These all are the parts of the ovule. Okay, so almost we'll discuss the collagen, nucellus, integuments, antipodal cells, embryo sac. This completely secondary nucleus, middle one. So the embryo sac will consist of the 8 nucleus and 7 celluled stage, Nana. So this is very important. Almost 7 celluled and 8 nucleus. So 1, 2, 3. This is the antipodal cells, 3, and synergids, 2 egg cell. So 3 plus 3. But in the middle one, the secondary nucleus in the 2n state. So that's why 8 nucleus and 7 celluled stage of the embryo sac that is presenting in the angiospermic ovule. So this is very important in an entrance point of view. Almost. So the embryo sac, that is the female gametophyte, the functional megaspore, almost in that. This is the 4 haploid cells, embryo sac mother cell, that is diploid, undergo the meiotic division, gives rise the 4 haploid cells. 4 will arranged in the linear state. So in that lower 3, in the 4 haploid cells, the lower 3 cells are degenerated and upper one. So that is the functional megaspore. So in that the functional megaspore, it represents the first cell of the female gametophyte. Okay. So in simple way, the upper one, the functional megaspore, represents the first cell of the female gametophyte. So mostly the collagen and grows along the micropylar collagen axis. So in this, the nucleus undergo the 3 mitotic divisions. The functional megaspore undergo the 3 mitotic divisions and form the 8 nuclei. One will gives the 2, 2, 2, 4, 4 to 8. So the nucleus undergo the 3 mitotic divisions and form the 8 nuclei. So in that 8, 3 will goes to the antipodal cells and 2 will synergids and 1 is the egg cell, remaining 2 is the secondary or polar nuclei. So just the nucleus, these nuclei are recognized, 1 nucleus from each group at a pole. So they will migrate to the center of the cell called the polar nuclei. So in simple way, first cell, it will undergo the 1 mitotic division, 2 will be formed. So 1 will go the micropyle side and 1 will come to the collagen side. Okay. So that is both are called as a polar nuclei. So the 3 nuclei, almost 1 will come to 4 to after 3 mitotic divisions. 4 will be present at antipodal anterior side and 4 will be posterior side. So in that 4, 1 will come to middle part of the embryo. So beginning upper one and lower one, they both will combine to form the secondary or polar nuclei. So same as per se, the 3 nuclei left at the collagen end, they are surrounded by walls and form the antipodals. So this is the antipodals. And 3 nuclei located at the micropylar end. So it constitute the egg apparatus. One serve as a egg or female gamete and other 2 as a synergids. So this is the synergid cell. This is the egg cell. The whole structure with 2 polar nuclei, 3 antipodals, 1 egg, 2 synergids. So that is the mature female gametophyte or embryo sac. So the mature female gametophyte is known as a embryo sac. Completely, the embryo sac consists of the 7-celled 8 nucleate stage of the female gametophyte. Okay, this is very important. So you can observe here, this is the almost all parts are see in this day. So the development of the female gametophyte, the female gametophyte first arises as a tiny protuberance from the placenta in the cavity of the ovary. So as it matures, the ovule consists of a nutritive cellular mass that is known as nucellus. You know that. So it protected by two integuments and attached to the placenta by a funiculus. Even at an early stage, one cell becomes evident in the nucellus. So that is known as the megaspore mother cell, simply MMC. Okay, almost in the center part, even at the primary stage, one cell, it becomes the evident in the nucellus. So that is the megaspore mother cell. It is in diploid state, 2N state, so 2X also. So it's almost for our convenience, MMC means megaspore mother cell is the diploid state. So then MMC increases in size and divides twice to form 4 megaspore as the linear tetrad. As per same, just you can observe the first diagram. The first division is the reduction division. The resultant megaspore are the haploid. Out of the 4 megaspore, 3 degenerate. It will forming the caps while one remains the functional. This functional megaspore is the haploid. So the functional megaspore then grows rapidly by absorbing the nutrition from nucellus and forms the embryo sac. The process of the formation of embryo sac, that is known as the megasporogenesis. Okay, the process of formation of the embryo sac. Embryo sac means almost 3 antipodals, 2 synergids, 1 egg cell and 2 secondary nucleus in the middle part. So the process of formation of the embryo sac, that is known as a megasporogenesis. So the embryo sac, it consists of 1 nucleus divided into 2 daughter nuclei, which move towards the poles. The pole of the embryo closer to the collagen. So as per same, almost each daughter nuclei divides again and result in the 4 nuclei. Each nuclei divides again. This is same as per. So that is the embryo sac. So it's nothing but female gametophyte. The types of the female gametophytes are distinguished by the number of megaspores involved in the formation of the embryo sac. So it is based on this criteria. There are 3 types of female gametophytes: monosporic, bisporic, and tetrasporic. So each type has more than one subtype. So some of the common ones are discussed further. Just I will show one diagram only. You can observe all these things. So in female gametophyte, monosporic, bisporic, and tetrasporic. Just what's happened? In monosporic, polygonum and enothera, bisporic, elysium and endomeon, tetrasporic. So number of types are there. So 7 subtypes. In that first is the fritillaria. Just this is the complete types of the embryo sac. Okay. So in that monosporic, bisporic, and tetrasporic. Just will take the types of the development of the embryo sac. Okay, the embryo sac in angiosperms, that is in 8 nucleate and 7 cellulose stage. We know that. So on the basis of number of megaspore nuclei which take part in the embryo sac development, 3 types are recognized. So monosporic type, bisporic type, and tetrasporic type. Just megaspore mother cell nana. So each cell undergo the meiotic division. After meiotic 2 division, 1 cell, 2 cells convert the 4 cells. So in that 4, 3 is degenerated. Only 1 will be functional. So the haploid functional cell undergo the continuous division to give a embryo sac. So that is almost the monosporic type. In this type, only one of the 4 megaspores formed from the meiotic division of the megaspore mother cell. It takes part in the development of the embryo sac. So this is the monosporic. This is known as a megasporic type. It is of 2 types. So in the 2, polygonum type and enothera type. Monosporic. So all the nuclei in the embryo sac are genetically identical as they develop from the same megaspore. There are 2 subtypes of the monosporic female gametophytes, polygonum type and enothere. So this is the polygonum. Just to simply, in the polygonum, the 8 nucleate monosporic type of the embryo sac, it was first discovered by Strasburger in Polygonum divaricatum. So in that way, it is known as a polygonum type. Okay. So the polygonum type embryo sac was first discovered by Strasburger in which plant? Polygonum divaricatum, and it is referred to as a polygonum type. So it is very common in angiosperms, and it is also called normal type of the 4 megaspores formed from the megaspore mother cell. So almost first megaspore mother cell undergo the first meiotic division. It will be gives the 2 cells. After second meiotic division, so gives the 4 cells. 4 megaspore formed from the megaspore mother cell. Only one is the functional, and it is participate in the development of the embryo sac. So the almost among the 4 haploid cells, among the 4 megaspores formed from the megaspore mother cell, in that only one is the functional, and it is participate in the development of the complete embryo sac. Okay. So in this, almost the collageal megaspore, the lower one. So you can see here, this is the remaining upper 3 is this is micropyle, this is the collageal side. The collageal megaspore is functional, and the remaining 3, 1, 2, 3, towards the micropyle end, the 3 will be degenerated. Degenerated megaspore, only one functional megaspore. Okay. So in this, the functional megaspore nucleus divide mitotically into the 2 nuclei.